Welcome to the session 1 video of our lab sessions for cell and tissue engineering. So the very first step is obviously to wear your PPE and be protected and sanitize everything that you're going to be using. And you can see here that that's being done. Everything is being sanitized. Just a disclaimer, in these videos, I won't mention numbers like mass, volume, temperature. Refer to your protocols to see what numbers need to be used when, let's say, volume is being pipetted or something like that. Here you can see that the first step is to thaw out a frozen vial of mesenchymal stem cells, and it helps to swirl the frozen vial around in a preheated water bath. And you want to be able to take this out around the point when you see ice crystals forming, and you can see that it has mostly thawed out here in the close-up view. It is very, very important to be careful but also move quickly, because the cryopreservative, which has DMSO, is a toxic agent when the cells are sitting in thawed out cryopreservative media. Here you can see that the flask in which the cells are going to be plated is being readied, and at the same time you can see that here the thawed out cells are going to be taken out and transferred into a conical tube. As you can see over here, carefully being put into a conical tube. And then to minimize the effect of the toxicity of the cryopreservative, we also want to make sure that we add fresh media into that conical tube to dilute the concentration of the toxic cryopreservative. So you can see here that fresh media is being added. And yes, we need a little bit more. And so at that point, we still have to move quickly, because remember, there's still that toxic cryopreservative in there, but it has been diluted significantly. So now we need to centrifuge it so that we can collect our mesenchymal stem cell pellet. The centrifuge has to be balanced, so that's why you can see that there was another tube on the other side of it. So again, refer to your protocol to get the specific numbers for the centrifuge setting. And you can see here that the centrifuge is being set up to spin. Under these conditions, a cell pellet will arise at the end of the centrifuge cycle. And so that will allow us to separate the cell pellet from that diluted but nonetheless toxic cryopreservative media that has been, again, diluted in fresh media. So with the centrifuge settings all in place, we can then proceed to have it start by pressing the start button, and you can see here it's all set to go, and it is spinning right now. So you want to be there on standby, again, in an effort to swiftly get the cells out. And so we can see here that the centrifuge cycle is approaching the end, and it's got a couple more seconds left before we can take our vial out of the centrifuge. So we want to be present there as soon as that marker hits 0, which means it's complete, and it would be advisable to take our tube right out at that point. Okay. So you can see the deceleration of the centrifuge to 0 RPM as well. And at this point, it is going to allow us to go ahead and recover our tube with the cells. So we can open the centrifuge up. We can then carefully take our tube of cells. And you can see here as a close-up, you can see a sort of a whitish, cloudy pellet at the bottom. Very clearly it is there. And that's our all valuable mesenchymal stem cells that we want to be able to now resuspend in fresh media. So once we've taken our centrifuge tube out of the centrifuge, we want to carefully but quickly move towards plating the cells, again, the mesenchymal stem cells, into a flask. The very first step is to remove that media that has the toxic cryopreservative that is also known as the supernatant, and we want to get rid of that. So we carefully remove the supernatant, and we get rid of it, and that can be disposed of carefully in a biohazard trash bag. The next step is to then resuspend the mesenchymal stem cells in fresh media. So we take an appropriate volume of fresh media and we gently mix the cells in there without creating air bubbles, because air bubbles can compromise the viability of the cells. So we carefully and gently resuspend the cells in the fresh media. And there we go. At this point, we're ready to plate our flask. And so we choose an appropriate size flask for the number of cells that we believe we have. We gently pipette the cells up, and then we gently pipette them out of the pipette tip into the flask. We do need additional media to plate so that the cells have sufficient nutrition to last them over the culture duration in the order of days. And so we take additional fresh media and then we pipette that into the flask, close the cap of our flask, make sure that a thin layer of cells suspended in fresh media is there over the entire flask. And last but not least, we move towards then carefully taking our plated flask of cells, mesenchymal stem cells, and placing that in the incubator at standard incubator conditions.